I want to first thank everyone for allowing us to participate in this challenge. This is the first time that I've been a part of this sort of event, and it's been a really interesting experience and one I've quite enjoyed, not just to hear what people have to say about our data, but really to see the social experiment that it is, which is to say, how do we all work together to address these very important questions, and how do we even collectively decide what are the most important questions to be spending our time on. So with that brief introduction, what I will do in the next few minutes is to describe to you the two NCI DREAM challenges, and then following my talk, Jim Costello will actually describe how the challenges were scored. And just a brief outline, I will actually really divide them first challenge 1 and then 2 to give a little bit of motivation about the challenge, the data sets available, and then to get into the nitty-gritty details. So the first challenge was to predict drug sensitivity in the panel of breast cancer cell lines that Joe just described. And as Dina mentioned, the data for this challenge are coming from Joe Gray's lab at OHSU. And what we have amassed in the lab over the course of many years is a very well-profiled panel of breast cancer cell lines. So here what I'm showing you is a Venn diagram where each circle represents one of these data sets, and we've profiled these cell lines transcriptionally, the exome-seq. Also, we've run exon arrays, pro bono methylation, and we have protein data and also genome copy number data. And finally, we have also tested these cell lines against many of the compounds that Joe described previously. And it's worth noting that several of these data sets have been published and are in the public domain, either in data repositories or in manuscripts. And additionally, we have also been depositing a lot of our data at the UC Santa Cruz Genome Browser with the intent that that is a way for biologists and everyday people to actually go in and try to play around with the data. So in an attempt to sort of remove the need for high-level computation. You know, someone can just go in and take a peek at some of the data available. What we have observed is, and what we do know, both from our breast cancer cell lines, but more importantly from primary tumors, is that the molecular diversity of breast cancer is huge, and that diversity makes it an incredibly difficult disease to treat. So what I'm showing you here is DNA copy number for our panel of cell lines, where here we have ordered the data along the genome from chromosome 1 to chromosome X. And what I'm showing here are changes in copy number. And what you can see is that across the cell line panel, there are a few key regions that are deregulated, either amplified or deleted, including amplifications at MYC, cyclin D1, and ERBB2. And these amplifications are things that are seen in primary tumors. If we profile the cell lines, here this particular data set is RNA-seq, what we can end up observing is that the cell line panel falls into many of the different clinically relevant subtypes, so the Claudin-Lows, basals, normal-like, and luminal cell lines. And these subtypes are important because they are clinically relevant. If you know the subtype, you have some inkling into what would be an appropriate treatment. However, it is the case that this is not the whole picture by any means. And all of these aberrations we need to account for to try to really understand how best to treat an individual patient. So just to give you a little bit of detail on the high throughput drug screen that we have run. For all of the cell lines, we've run this standard biological assay where we treat the cells for 72 hours with one of these therapeutic compounds, and then we measure viability with a CTG assay to count the number of cells that are still living after 3 days of drug treatment. And from those data, what we can end up doing is computing a GI50, or a dose response curve. So here, what we're plotting along the X is drug concentration from low to high. And along the Y, what we are doing is plotting growth inhibition, which essentially is the number of cells still living after 3 days of treatment. And what you can see is that at low drug concentrations, there are a lot of cells left. after we reach some critical drug concentration, we end up seeing fewer and fewer cells after 3 days of treatment. And we typically see a sort of plateau. And this is the prototypical drug response curve. What we have found after looking at thousands of growth response curves is that there actually is some variation in what the curve looks like. But this is the general trend we see. And across the cell line panel, what we see is that there's quite a variation in response to individual compounds. So in this bar plot, each bar represents one of our breast cancer cell lines, where the height of the bar represents how sensitive that cell line is to a compound. In this case, it's an AKT1/2 inhibitor. And what you can see is that some of the cell lines are quite responsive, and other cell lines are much less responsive to that compound. And what we have tested are over 140 compounds now, a subset of which we have put into DREAM. But just to give an idea, for this particular DREAM challenge, we actually held back what the drug target was, with the idea of making it that much more challenging for you all to try to solve the problem. But this just gives you an idea of some of the targets that we have been looking at. So some of them are sort of more classic chemotherapeutics, but many of them are these targeted therapies that are really designed to go in and with a laser, hopefully with a laser, go in and knock down one particular node in a network, for instance. And it is the case that these different compounds show a quite different variation in dynamic range. So on this box plot, set of box plots, what I'm showing you is the dynamic range for each compound in the DREAM challenge. And they're ordered by the median GI50 value. And what you can see is that some compounds show a relatively narrow dynamic range, whereas other compounds show a much wider dynamic range. And it's these compounds with a wider dynamic range that typically are easier to identify a predictor for, although it turns out that in our hands that's not 100% indicative of how easy it might be to generate a predictor. But nonetheless, it is an important consideration, and one which was considered when we were scoring the different challenges as well. So the challenge in schematic format was to develop predictors of drug response for the data I just described. And the details of the challenge are illustrated here. So the input data is all of the genomic data for 6 genomic data sets. So copy number data, protein data, methylation, exome-seq, gene expression from arrays, and RNA-seq data. And participants were able to access data for 53 cell lines. They then were given drug response data for 35 of these cell lines, and 18 of the cell lines were held out. And the challenge was to try to determine, based on the training data, what the drug response would be for the test data set. And what we asked in detail to get from our participants was a ranked order list of all of the cell lines. So how well did each of the cell lines in the training and test set respond to these compounds? All right, so for sub-challenge 2 was a prediction of synergy. So in all of the work I just described, what we are considering is the single drug case. But it's becoming more apparent that perhaps using dual therapies could actually be quite beneficial for treatment. But there is a little bit of a challenge in determining what the best drug pairs are. So this schematic that I have taken from a recent review shows, for illustrative purposes, cell signaling in the MAP kinase, PI3 kinase pathway. And I put it up here just to make a few points, which first is that there's a lot of crosstalk between signaling nodes in this network. There's also a lot of feedback, and there's also further a lot of redundancy. There are a lot of ways to get to these different endpoints, which is, of course, very important from the cell's point of view to try to maintain homeostasis. But from a treatment point of view, poses a very big challenge, because if we end up treating a particular node here, we may affect how signaling in a different part of the network responds. So if we can somehow figure out what two parts of the network may be best to target, then we may have more effective treatments, and it's also possible that we could be treating at lower doses, which would lead to a reduction in off-target effects and maybe even a reduction in side effects. So when we're talking about applying multiple drugs, we are thinking about our complex drug interactions, such as synergy and antagonism, which I have schematized here in a little cartoon, where here what I'm showing you are 3 drug response curves, where again we're plotting drug concentration along the x, so from low to high concentration, and again the number of viable cells following treatment. And if we, for instance, have one particular drug A, we may see a slight drug response. A second drug B may give a better response, but the combination of the two may yield an even better response still, which is to say that we've shifted the curve to the left, so a lower concentration is required to achieve a response in the cells. The opposite effect is antagonism, where if I, with the magic of PowerPoint, simply change the colors of the curves and now consider orange and blue to be the single treatment and green line to be the double treatment, then what we're seeing here is a shift toward a less effective treatment. So the problem, as I've been alluding to, is to try to determine which drug combinations will be synergistic or antagonistic. And for this challenge, what we've had is an extensive survey of LY3, which is a diffuse large B-cell lymphoma. And the data available for the challenge are a set of drug response data, so IC20 concentrations for 14 compounds, and those IC20 calculations were computed after either 24 hours or 48 hours. And additionally, we also provided the raw drug response data underlying these calculations. The second part of the data available was a very extensive profiling data set, where we provided 252 treatment gene expression arrays from an Affymetrix high throughput platform. And in these arrays, this cell line LY3 was subjected to treatment with one of the 14 compounds at these 2 concentrations detailed on the previous slide at one of 3 time points, 6, 12, or 24 hours. There were also 30 control gene expression arrays, the untreated arrays, and finally also baseline genetic information from the Affy-SNP 6.0 chip. So the challenge here was to use the post-treatment transcriptional profiling data to predict whether compound pairs will be synergistic, additive, or antagonistic. So as I mentioned, the input data are all of these transcriptional data, and the prediction, the challenge was to tell us which of these combinations would lead to a different interaction, and to return to us a ranked list of all 91 compound pairs, from the most synergistic to the most antagonistic. In order to score the challenge, what we needed to do was to calculate a gold standard and to actually experimentally assay a measurement of synergy. So to do this, in the past few months, there has been a flurry of work to test these 91 drug combinations at the IC20 concentrations I detailed previously, and we have actually computed synergy for each drug pair. And here is just a quick schematic of how that assay was run. And there are many different ways of computing synergy, and what we chose here for this challenge was to use this Bliss additivism model, where the formula is here, where A and B are the fractional response of the individual compounds. And then we compute the excess over Bliss, Z minus C, where Z is the fractional inhibition induced by combination of A and B. And this EOB is the measure of synergy or antagonism. And with that, I would just like to acknowledge the major participants of the 2 challenges, roughly organized by Challenge 1 and Challenge 2, although really there has been a lot of interaction among the entire group over the past few months. Thank you. And with that, I will hand off to Jim.
Are there any questions for Laura? Okay, in that case, Laura told you about the data that were used. Jim Costello from BU is now going to tell you about